Okay, now we're going to talk about lipids. Lipids. When you think lipids, the one thing you should think of is hydrophobic. They are hydrophobic, meaning they do not like to interact with water. All right, so let me share my screen. iPad. Okie dokie. All right. So I apologize, these slides are a little busy. If you download the actual PowerPoint, which I've posted, they're all animated so you can bring things in individually. And I highly encourage everyone to start looking at that. So lipids are a diverse group of hydrophobic molecules. You're gonna see me using this abbreviation a lot. Water unhappy or water fearing, right? For hydrophobic. So these lipids are a group of biological molecules containing regions composed almost entirely of carbon and hydrogen linked. So by what kind of bonds? What kind of covalent bonds link, for example, a carbon and a hydrogen? What kind of covalent bond is that, right? That is going to be a non-polar covalent bond, just like the same bond between a carbon-carbon molecule. Those are both non-polar covalent bonds. All right. They have a huge variety of functions. They are important in energy storage as well, just like carbohydrates. And this is in the form of fats and oils. They are also involved in waterproofing. So you can see that, for example, on the cuticle of a leaf, which helps it be waterproofing. We're going to talk ad nauseum about the lipids that are involved in cellular membranes, which are over here. These are called the phospholipids, right? So phospholipids. These are very, very important for cells. I mean, they come together in very interesting ways based on their polar and non-polar parts. But again, it's a lipid, so it has a lot of hydrophobic regions. And then the last one, which is kind of confusing for some people, are called steroids, right? And we're going to talk about hormones and steroids a lot later on in the class. But steroids here, down here, are composed of 1, 2, 3, 4 carbon rings when they're put into a watery environment. All right, so when we talk about lipids, they're in 3 major groups. Number 1, we have oils, fats, and waxes. And really, we don't talk a ton about waxes. Really, we're talking mostly about oils and fats. Number 2, phospholipids. And these, again, are very important in cellular membranes. And then number 3, steroids, which are these 1, 2, 3, 4 carbon rings. And those are going to be really important for cellular signaling when we get there. So let's talk about those groups first. So the oils, fats, and waxes are made primarily of fatty acids, right? So fatty acids. So what is a fatty acid? It's got kind of a funny name. So a fatty acid, by definition, is a carboxyl group, right? So again, here's our carboxyl group. These are why we have to know all of those functional groups. So here is our kind of COOH carboxyl group hooked to a hydrocarbon chain. And a hydrocarbon chain is a bunch of carbons fully kind of with a lot of kind of saturated or bound to a lot of hydrogens. Remember, every single one of these little bonds, these are covalent bonds, but more specifically, they are non-polar covalent bonds, making them very hydrophobic, right? Which we know lipids are. However, you do realize that we have a kind of, we have this, oops, let me go back there. We have this carboxyl group. And you're right, if you're looking at that, you're like, that is in fact polar. But in general, it is mostly fatty acids, all of this, making it mostly non-polar. Okay, so when we look at oils, fats, waxes, etc., they consist mostly of carbon and hydrogen atoms linked together by these covalent non-polar bonds. Fats and oils are for energy storage. They, in particular, fats, at least 2 times as many calories per gram as carbohydrates or proteins. So they're really packed with high energy, or high energy storage. And then we also have the waxes, where they're important for waterproofing. I'm not as concerned about waxes. They come in different, multiple varieties, right? So first here we have the fats. This is called a triglyceride, right? And the reason it's called a triglyceride is that you have one glycerol molecule, which is here, right? One glycerol molecule, and 3 fatty acid chains, right? So a triglyceride has 1 glycerol and 3 fatty acids. So when I ask you on your biological molecules sheet to kind of draw out a lipid, this is the kind of thing I want you to know. So instead of having traditional monomers like the kind of carbohydrates that we were talking about, the monomers for lipids are glycerol, 3 fatty acids, for example, for a fat. For a wax, it's different. It's an alcohol and a fatty acid. And then fatty acids, right? Those are kind of different monomers. All right, so let's go over here again. So we have 1 glycerol molecule. We have 3 fatty acids. So how many dehydration synthesis reactions did we have there? So we have 1, 2, 3. So we're going to get the release of 3 waters out. And now we have the formation of a single fat, or a triglyceride molecule. All right. So here is an example down here of waxes. I'm not even going to go over this, but you should understand that you should be able to recognize the fatty acid because you have your carboxyl group, and then you have this big, long fatty acid, hydrocarbon chain. So this is your hydrocarbon chain there. And you can see there's 14 of these. So it's a lot of those coming together. And that's how you make a wax. So fatty acids come in 2 main flavors, if you will. You can have saturated fatty acids, which mean they are solid at room temperature. And the way I always think about that is butter is solid at room temperature, and therefore it is a saturated kind of fat. That's how I remember it. And unsaturated are oils, and those are liquid at room temperature. So if we look at the fatty acids in, for example, an oil versus if we look at the fatty acids in a saturated versus an unsaturated, they look fundamentally different. So saturated, what that means is that they are saturated with hydrogens. So every carbon on that fatty acid tail has the full amount of hydrogens possible, right? So it is saturated with hydrogens. The unsaturated ones here in the oils are unsaturated. And what I mean by that are that there are double bonds. And what a double bond does to a fatty acid tail is that it puts a kink in them, right? And so every time you see kind of a region that is where the tails are kinked, that means that there is a double bond in the chain, which causes the skeleton to kind of kink. And the way I remember that, and apologize, is maybe a little bit inappropriate, but the oils are kinky. Kinky oils, unsaturated, meaning you have these double bonds. Okay, the second main group are called the phospholipids. These form plasma membranes around every living cell, right? They're the major components of cell membranes. They're similar to fats. And if you remember, a fat is a triglyceride, and that was made from one glycerol plus how many fatty acids? 3 fatty acids. A phospholipid is basically very similar. Instead of one of the fatty acids, we're going to put a phosphate group there, right? So here is our glycerol molecule. So that's our glycerol molecule. We're going to have, so we have 1 glycerol. We're going to have 2 fatty acid tails. And then on the third kind of reaction, we have what's called a phosphate head, right? That's my abbreviation for phosphate, is kind of this P with a circle around it. That's your phosphate group. And you should know that because that's one of your functional groups. All right, so the way that we draw in cartoon form a phospholipid is kind of like this, all right? Oops, here I'm gonna draw it somewhere where you can see it a little better. I'll draw it here. So you're gonna draw a circle that is going to represent the phosphate head. Then you're gonna have 2 fatty acid tails. Notice how I drew them. I drew one straight and one with a kink in it, meaning that it is fundamentally different because it has a hydrogen bond. Sorry, a double bond. All right, so this head is polar. And the reason it is polar is because of that phosphate group. We know the functional groups, we know phosphate's polar, a polar molecule. The tails, we have 1 fatty acid that is saturated here, and we have 1 fatty acid that has a double bond, and so that is unsaturated, right? So all phospholipids have a polar head and a non-polar tail. Polar head, non-polar tail. So phospholipids have these water-soluble, or let me rephrase, water-loving or hydrophilic head, and water-insoluble or hydrophobic tails, right? So do you remember what it's called when you have a molecule that has both polar and non-polar parts? That is again called amphipathic, right? It's an amphipathic molecule, both polar and non-polar parts. Phospholipids have this glycerol backbone. They have a polar head with a phosphate group, and then they have 2 fatty acid tails that are non-polar. Usually one is saturated with hydrogens and one is unsaturated because it has a hydrogen bond, right? They also have these variable functional groups. Don't really care about them except that they're polar, right? So again, if we draw this out, we have polar head, non-polar fatty acid tails. All right, we're going to go over this in class together. The third main type of lipid is a steroid. And these, again, contain 4 fused carbon rings, all right? The basic structure of all steroids is cholesterol. And we've been over that before, right? We've looked at that before. So it has 1, 2, 3, 4 fused carbon rings. If you look at this structure, all of this is polar or non-polar. It's going to be non-polar. Yes, it has this, what is this functional group? That's hydroxyl, that is polar, but most of it is non-polar, which is great because we're talking about lipids. And we're going to talk a lot about how these, oh my God, hi there, Will Ferrell. He's saying, when you think of lipids, you should think non-polar. Anyway, so they're the basis of all steroids is this carbohydrate, and then they can have different kind of functional groups. So for example, what is this functional group? CH3. You should know that. That is methyl, and you should know polar, non-polar. You should know that's non-polar. Here's another one. We have hydroxyl. You should know that's polar, and that is hydroxyl. All right, and we're going to talk later when we get into membrane transport how steroids are special because they can slip right through the plasma membrane, which is made again of these little molecules that we just talked about with a polar head and a nonpolar tail, phospholipids, right? And steroids can go right through them. And we'll talk about that later. And that's it for lipids for now.